Hello students. In the previous lecture, we had seen enteric pathogens. In that E. coli we had started with respect to classification, biochemical characters, antigenic structure, viability characters. Okay. Now continuing E. coli, we will move ahead to its pathogenicity, pathogenesis, symptoms, laboratory diagnosis, epidemiology, prophylaxis, treatment. Okay. Now first, let me tell you what exactly these words are. Pathogenicity. The property of causing disease is called as pathogenicity. Pathogenesis, the manner of development of a disease. Okay? Symptoms. Whatever the clinical symptoms are seen when a person is infected with some or the other infection. Okay? Laboratory diagnosis. In this, we will see what tests are conducted, how the organism is identified. Okay. Epidemiology. In epidemiology, I told that the disease, the scientific study of the disease is done. Okay. Next, prophylaxis. Treatment given or action taken to prevent the disease. It is called as prophylaxis. And last is treatment. Okay. Let's begin. First, pathogenicity. Now there are some factors which are involved. We can also call them as determinants of pathogenicity. Okay. First, adhesion factor. Second, enterotoxins. Third, enterohemorrhagic E. coli. Fourth is antiphagocytic capsule. First, adhesion factor. Now, strains of E. coli, they are responsible for urinary tract infections, and they have unique AFA1, AFA3 and Dr. adhesins. Okay, that allow adhesions of E. coli to the epithelia of calyx and collecting ducts. It's their role. Fine. E. coli involved in the lower UTI have specific O antigens which are believed to allow adhesions to the bladder epithelium. See, it's a pathogenicity part. Means these are the properties which are causing disease. So first we saw was adhesion factor. Second, enterotoxin. Now in enterotoxins, some strains of E. coli, they produce enterotoxins that cause gastrointestinal disorders. Now enterotoxin toxigenic strains of E. coli, we refer it as ETEC. Okay. They produce either one or two plasmid mediated enterotoxins which are heat labile and heat stable enterotoxin, means LT and ST. Fine, I have mentioned it over here. Now heat labile enterotoxins, LT. Now they are protein and resemble the cholera toxin CT in all respects, such as structure, antigenic property and mode of action. However, CT is about 100 times more potent than LT. LT can be detected by several serological tests. Okay. Next, heat stable enterotoxin ST. In contrast to LT, heat labile enterotoxin, ST is a low molecular weight polypeptide and poorly immunogenic. Two major classes of E. coli STs are recognized, STA and STB. Both are plasmid encoded. STA binds to cellular guanylate cyclase and cause activation of guanylate cyclase activity, leading to increased formation of cyclic guanosine monophosphate cGMP within the epithelial cells, which in turn deregulates iron transporters and can cause effects similar to those of heat labile enterotoxins. Okay. Next, enterohemorrhagic virotoxin producing E. coli. See, it is labeled as EHEC or VTEC. Understood? Enterohemorrhagic E. coli, EHEC, virotoxin producing E. coli, VTEC. Now, it was so named because of its cytotoxic effects on vero cells, a cell line derived from African green monkey kidney cells. Okay. Now, verocytotoxin is also called as Shiga toxin, Shiga-like toxin, SLT, due to similar biological, physical, and antigenic properties of the toxins. Now, Shiga toxins, they are produced by strains of Shigella dysentery type 1. We will look at this point later when we will see Shigella. Okay. Now, the most virulent uropathogenic strains of E. coli produce hemolysins, which can lyse RBCs of some species and have general cytotoxic properties. Understood? Antiphagocytic capsule. Now, the K1 strain of E. coli is a major cause of neonatal meningitis. Okay, 80%, which has antiphagocytic capsule. K1 celiac acid homopolymer capsules are cross-reactive with human adhesion molecule, which is NCAM. Okay. Newborns and infants being tolerant to this NCAM. Now, NCAM is what? Neural cell adhesion molecule. Now, newborns and infants are tolerant to this NCAM, which probably causes a lack of reactivity against K1 capsules. Understood? These are the determinants of pathogenicity which are involved in disease causing. Okay. Next, we will see symptoms. What are the symptoms when a person is infected with E. coli? Abdominal cramping, sudden severe watery diarrhea that may change to bloody stool also, gas formation, loss of appetite or nausea, vomiting. It is uncommon. Okay. Fatigue, fever. Now, clinical infections caused by E. coli. These are the symptoms. Now, infections: urinary tract infection, septic infection of wound, diarrhea, dysentery, septicemia, pneumonia, neonatal meningitis, and abscess in a variety of organs. So, E. coli will show these symptoms and clinical infections caused by E. coli. Symptoms can be similar with other type of infections also. So it is not just that abdominal cramping is there or vomiting is there or some diarrhea is there. So it doesn't tells you that that the person is infected with E. coli. No. These are the symptoms, but on laboratory testings, on diagnosis only, it is confirmed that the infection is caused by E. coli. Okay. Next, pathogenesis. Now, we will see the manner of development of disease. Now, pathogenic E. coli with each pathotype may be further classified as virotypes based on the virulence genes that they possess. We have discussed the determinants of pathogenicity. So now we can understand what classification is done. A virotype is a particular combination of virulence genes. Important virulence factors encoded by these genes include fimbrial and adhesins, enterotoxins, cytotoxins, capsule, and lipopolysaccharide. Okay. Pathogenic E. coli may also be differentiated by serotypic based on antigenic difference like O antigen of the LPS, flagellar or H antigen, or the fimbrial or F antigens. We have seen them and what is their role. Fine. See, ETEC, what it stands for? Enterotoxigenic strains of E. coli, ETEC. This image will show you how the pathogen is working. The pathogenic food is injected which is having E. coli. So after that colonization will be formed in intestine or jejunum or ileum. Then in the epithelial cells some receptors, toxins and fimbriae of E. coli, they will act. They will release waters and electrolytes, dehydrates the body in the form of diarrhea, which will ultimately give impacts to weight loss, or it can be fatal also if not treated. Understood how it is working? Now, pathogenic bacteria contaminating the environment are ingested by susceptible animal as shown in the figure, and they enter the intestinal tract. Now these bacteria, they possess fimbrial adhesins which mediate adherence to specific receptors on the intestinal epithelial cells. Understood? This image you can see over here. Resulting bacterial colonization is found mostly on the jejunal and or ileal mucosa. Now the adherent bacteria produce enterotoxins which stimulates water and electrolyte loss into the intestinal lumen. Now because of that, as I told you that dehydration happens, and if it is not treated it can be fatal also, and you can see that the weight of a person is decreasing. Okay. Now two steps we have seen over here: intestinal colonization and hypersecretion of water and electrolytes. Understood the pathogenesis, ETEC? Next, EPEC. Now what is EPEC? Can you tell me? It is enteropathogenic E. coli, EPEC. Now here you can see the same image, but there's a difference where in colonization occurs in the small and the large intestine. Now here what happens? See, here you can see bacteria produce their own specific receptors. There's a change. There's a change. Bacteria produce their own specific receptor which is injected into the host epithelial cell via a syringe-like bacterial apparatus. Here you can see a syringe-like apparatus. See? Okay. Now a bacterial adhesin then mediates a very intimate attachment of the bacteria to the cell receptors, and they give signals to stimulate effacement of the microvilli or the brush border and reorganization of the cell cytoskeleton. Understood what is happening? Now, the adherent bacteria also stimulates epithelial cell degeneration and infiltration of PMN in lamina propria. So, this is what the pathogenesis is going on with EPEC. These cells changes may lead to the appearance of diarrhea. Okay. Next, EHEC or VTEC. As I told you, EHEC is enterohemorrhagic E. coli, VTEC, verotoxigenic E. coli. Understood? What happens? Now, most common strains producing gastrointestinal disease. Okay, we have already gone through this. And it can cause common diarrhea and hemorrhagic colitis. Understood? Fourth is septicemia and pyogenic infections. Okay. Now, what happens in this? Septicemia is caused by E. coli and other some gram-negative bacilli, which originates from infections in the urinary or gastrointestinal tract. For example, intra-abdominal abscess or some intestinal perforations are there that time. And E. coli can cause wound infection, peritonitis, biliary tract infection, and neonatal meningitis. We have already seen. Okay. Neonates and prone to infection due to lack of IgM antibodies, E. coli with group B streptococci are most important causes of neonatal meningitis. 75% of these strains contain K1 capsular antigen. We have already seen K1 capsular antigen. Neonatal meningitis is acquired either during birth or postnatally. Okay. Epidemiology. Fecal oral transmission, foodborne transmission, and endogenous infection. Fecal-oral transmission. Transmission of E. coli occurs from person to person, usually by fecal-oral route. Next, foodborne transmission of E. coli is observed in cases of severe gastroenteritis caused by type O antigen 15 and H antigen 7, traced to contaminate meat. This is a foodborne transmission. Third is endogenous infection of E. coli, which is observed in urinary tract infection from the perineum. In women, UTI is frequently associated with sexual activity, hence the condition is also called as cystitis, honeymoon cystitis. Okay. Laboratory diagnosis. Now, what all is done? Sample collection, microscopy, and culture. Sample must be collected depending upon the site of lesion. Now, for urinary tract infection, urine. Now, for acute diarrhea, feces and rectal swab. Wound, pus is collected, and pyogenic meningitis, CSF is collected. Okay. Moving ahead, microscopy. Gram staining is done. In that, we can see that the bacilli is a gram-negative bacilli in background of a large number of pus cells. This is the microscopy. And what happens in the cultures? Macconchis agar, E. coli, it grows, showing lactose fermenting mucoid pink colored colonies. In blood agar, moist non-hemolytic or creamy white colonies are seen. Nutrient agar, colorless, smooth, large colonies are seen. See, you can see over here E. coli growth on different type of agar plates. Macconchis, blood, nutrient. Understood? Prophylaxis. Now, what are the actions we need to take to prevent this disease? Practicing safe food behaviors first. Now, in this, what we can do is washing fruits and vegetables thoroughly, avoiding cross contamination by using clean utensils, pans, and serving platters. Okay. Keeping raw meats away from other foods and away from other clean items. See, these are the sources from where the infection can be spread. Contaminated water, food, meat, sewage, anything. Okay, so what practice we should follow? That we should not be infected by E. coli. So these are the steps. Then refrigerating leftovers immediately. Drinking only pasteurized milk products. Avoiding raw milk. Means you have to drink boiled milk or pasteurized milk. Understood? Not preparing food if you have diarrhea. It means if XYZ person has diarrhea, he should not prepare any food, or he should not touch the utensils, so that no other person is getting infected because of him. Okay. One of the easiest thing you can do to prevent an E. coli infection is to regularly wash your hands. Students, in this pandemic, we have learned a lot of hygienic things which we should follow our whole life. Washing hands, wearing mask, while handling some stuff, we should be careful. Okay. So similarly, here also, regularly wash your hands. You should wash your hands before handling, serving, or eating food, and especially after touching animals, working in animal environments, or using the bathroom. Practicing good hygiene and following food safety guidelines can go a long way to decreasing your risk of infection. Now, this safety is for all other infections, so that you don't get infected. So, always practice for good hygiene. Got it? This is not only for your study part but for your day-to-day life. Okay? Treatment for E. coli infection. Now, the only way your doctor can know that yes, you have an E. coli infection is to collect the sample of your stool and send it to lab for analysis. I told you before, just because of the symptoms, you cannot tell that you have an E. coli infection or you have a Shigella infection. Okay? So, your doctor will collect your stool sample and send it to lab for analysis. Fortunately, the infection usually goes away on its own. So, it's on the immunity level. Okay? For some type of E. coli associated with diarrhea, such as the watery traveler's diarrhea, antibiotics can shorten the length of time you have symptoms. What will antibiotics do? They will suppress the symptoms and might be used in moderately severe cases. Okay? But if you have fever or bloody diarrhea, and if your doctor suspects Shiga toxin producing E. coli, antibiotics should not be taken. Now, these all things are the concern part for doctors. They will decide what is to be done. It is not us to decide what the treatment should be given, or we should take for ourselves. Okay? Now, it's important to rest and get plenty of fluids to replace what your body is losing through vomiting or diarrhea. Okay, when you're dehydrating, it is simple that you have to uh, get plenty of fluids to replace the dehydrated thing and to take rest. Because of dehydration, you will be getting lot of weakness. Understood? Don't take over-the-counter medications that fight diarrhea. Means, as I told you, that anything which is happening to your body or your health issues, never take own treatment. Always go to a doctor, take prescribed medicines. Never just go to a chemist directly and ask for a medication, that I have diarrhea and I need some medication, because we don't know that diarrhea is because of which infection. It will just suppress your diarrhea, it will slow down your digestive system. Understood? And it will finally, it will delay your body's shedding of that infection. Means whatever infections your body is carrying, it will not shed off so easily, it will just get suppressed, your diarrhea will stop. So never take any medication on your own, always consult a doctor. Fine? Understood? If you have any doubts regarding E. coli, do ask me. I will upload the notes on Google Classroom. Fine? Thank you.